Dr. Saab, namaskar. Dr. Ramanathan, namaskar.
Doctor,
How are you?
Sir. How are you, sir?
How are you, Dr. Saab?
Sir?
Everything is fine?
Yes, sir. Everything is fine. How are you, sir?
Everything is fine, Dr. Saab.
Sir, good afternoon, sir.
Good afternoon, Dr. Saab.
Good afternoon, sir. This is Srinivasu, sir, Associate Dean from College of Horticulture.
Yes, thank you, Dr. So nice of you. So everything is fine, Dr. Saab?
Yes, sir. Yes, sir. Yes, sir.
How is the COVID situation?
Sir, here the cases are more, but we are in safe conditions, sir. There is no problem, sir.
You all should be fine. You all should be fine.
Yes, yes, sir. Yes, sir. How about, sir, the situation there?
Nagpur, yesterday 3,500 positive case, but fortunately, God's grace, situation is under control. Their infection is there, but it's okay. Not that much serious as it was last year.
Yes, yes, sir. Yes. Same is the case here also, sir, in AP also. Though the number of cases are positive, I think the cases are more. But the situation is not alarming as in the second wave.
Oh, good. Right.
Sir, we'll wait for 5 more minutes, sir, and start the program.
Okay. I join after 5 minutes. No problem. I'm here only.
Yes, sir. Yes.
वही नेचुरल फार्मिंग की कुछ चिट्ठी आई थी ऐसे
नेचुरल फार्मिंग का अभी अपने को जो मंथली कैपिटल रिपोर्ट भेज रहे हैं ना उसमें वो कंपोनेंट ऐड करना पड़ेगा
बट वी हैव टेकन एक्शन इमीडिएटली ब्लॉक नंबर 6 7
नेक्स्ट मंथ में अपन दिखाएंगे व्हाट सब एक्टिविटी वी विल हैव टू शो
Ramana,
Sinu, చెప్పు.
Ramana, shall we start?
Yeah.
Huh?
34, you can start now.
You can start, na? You can start the program.
Yeah.
Because sir may be having another,
Yes,
so we
yes.
can start the program. So, good afternoon, sir.
Good afternoon.
Good afternoon, sir. Sir, on behalf of Honorable Vice Chancellor Dr. Y.S.R. Horticulture University, all university officers, faculty, and students, particularly who are in final year of BSc Honors Horticulture, and also agriculture students who are participating in this program, and on my own behalf, I extend hearty welcome to you, sir, for this Citrus Graduate Readiness Program. So this is the 21st guest lecture in a series of 30 guest lectures planned for the Citrus Graduate Readiness Program. And we are highly thankful to you, sir, for your presence in the inaugural day of this Citrus Graduate Readiness Program, which was organized on 8th December 2021, that is last year. So since then, the program is being continued without any hurdles in spite of this COVID-19 pandemic situation. And with all your good wishes, sir, the program is being continued without any obstacles. And the students have been actively participating in this program, not only from Dr. Y.S.R. Horticulture University, from other agriculture and horticulture universities, students have been actively participating in this program. And whatever the guest lectures so far covered were highly useful and beneficial to the students. And the students have been a lot of interest. Even they have participated in 2 quiz programs, which were organized for the students. So one at the end of the 10th lecture, and the second quiz program was organized at the end of the 20th guest lecture. So students have been actually participated in those 2 quizzes also. And today's program is 21st guest lecture, sir. And sir, as you are already aware that this is one of the flagship programs organized by Dr. Y.S.R. Horticulture University, particularly for the students who are in ELP, Experience Learning Program, and also Rural Horticulture Work Experience Program, to enrich their knowledge in citrus crop, as this citrus crop is one of the important commercial fruit crops that is being grown in AP as well as in other southern states. So in order to have the full-fledged information and latest information on this crop, so eminent speakers have been invited for this program, and they are also participating in this program. And we are also thankful for the participants so far. They have given the guest lectures on this Citrus Graduate Readiness Program, sir. So with this few introductory remarks, I once again welcome you, sir, for this Citrus Graduate Readiness Program of 21st guest lecture. And may I now request Dr. K.T. Venkatramana, Professor Horticulture and Head of Horticulture Research Station, Anantarajapeta, to kindly take over today's program and extend welcome to our today's guest speaker, Dr. Dilip.K.Gosh, the Director, CCRI Nagpur. May I now request Dr. Ramana to kindly proceed further with this program.
Thank you, Sinu. Good afternoon to one and all. Today, as part of Citrus Graduate Readiness Program, we are privileged to have with us very distinguished speaker, Dr. Dilip.K.Gosh, the Director of Central Citrus Research Institute, Nagpur. On this occasion, it gives me a pleasure in giving introductory remarks about Dr. D.K.Gosh, Director of esteemed National Institute for Citrus. After completing his master's and doctorate degree from IRI, Pusa, New Delhi. Dr. Ghosh joined CCRI Nagpur as scientist in 1995. During last 26 years, he has made valuable scientific contributions for the citrus industry of the country by developing rapid and sensitive disease diagnostic tools that have been successfully used to implement citrus budroot certification program. More than 45 lakhs disease-free nursery plants have been developed at this CCRI Nagpur for the citrus growers of the country. So far, as a member of the multidisciplinary team of scientists, Dr. Ghosh was involved in the development and release of 5 citrus varieties at CCRI Nagpur, namely Qatar Valencia, US Pomelo 145, Flame Grapefruit, Nagpur Mandarin Seedless 4, and NRCC Pomelo 5 for commercial cultivation. Recently, Dr. Ghosh has developed user-friendly and low-cost rapid diagnostic tools for citrus greening disease and citrus tristigia virus. He has also first time reported the infection of Candidatus phytoplasma, Cynodontis, and hop stunt viroid of citrus. A widely traveled professional, Dr. Ghosh worked as a visiting scientist twice at the University of Florida, USA, visited many citrus growing countries like Brazil, China, Vietnam, Thailand, to share his research findings at various international forums. As an international FAO consultant, Dr. Ghosh worked for 2 months in Nepal on citrus regeneration and for a week in Bhutan. He also organized 3 international training programs for the scientists from Oman, Bhutan, and Nepal at Nagpur that was funded by respective foreign governments. In collaboration with IIT Roorkee and University of Central Florida, USA, Dr. Ghosh recently identified and demonstrated the efficacy of antimicrobial 2S albumin protein obtained from pumpkin seeds to inhibit citrus greening disease and jointly published 1 US patent. A fellow of 4 different scientific societies, Dr. Ghosh was also awarded prestigious Professor J.F. Dastur Memorial Award of Indian Phytopathological Society in 2020. He was instrumental in bringing several externally funded research projects from DST, DBT, ICAR, MIDH, and guided 6 PhD students, and also published his work in reputed international journals, including his recent publication in scientific reports, that is Nature. With this brief introduction, I welcome you, sir, for the presentation, and I'm sure it will update the knowledge of students and faculty on the subject. Thank you. Now I request, sir, please, you can make your presentation, start a presentation, sir.
Yes. So, slides here. So, thank you, Dr. Ramannaji. And I initially must thank the university, Dr. Y.S. Horticulture University, Honorable Vice Chancellor, Dr. Janki Ramji, and Dr. Srinivasuluji, and for inviting me to give today's presentation. And my sincere thanks to Professor K.T. Ramannaji, the head horticulture research station of Tirupati, and who happens to my very good friend from IRI Our Life. I remember that we spent time together while we are at IRI. Thank you very much, Dr. Ramannaji, for this invitation. And in my presentation, today's presentation, I'll talk about diagnostic characterization and management of virus and virus-like diseases of citrus. And the points which I'll include in my presentation, that is briefly about citrus virology, the developing diagnostic tools, targeting the major citrus pathogens, which is virus-like pathogens, its molecular characterization, and management that includes both conventional as well as biotechnological approaches for managing this group of pathogens. As you know, the history of plant virology is just about 120 years old, and the number of plant viruses reported to infect the different crop plants, it was around 89 during the year 1939. And since the development of science, technology, diagnostic techniques, now it is more than 1200 the plant virus that has been reported to infect different crop plants, whether it is a horticulture crops or field crops. As far as the horticulture crops are concerned, you see there's nearly 60% of the pathogens, that is virus, viroid, phytoplasma. These are the all graft transmissible pathogens. These infect different horticulture crops. And the crop like citrus, which is vegetatively propagated crop, where there is every chance of spreading the disease from nursery to the planting bed, because it's a horticulturally a vegetatively propagated crop, the seriousness is very, very important. As far as the citrus is concerned, you know, there are more than 50 virus and virus-like pathogens has been reported to infect citrus crop. That includes at least 5 viroids, a phytoplasma, a spiroplasma, 3 different Liberibacter species that causes the dreaded citrus greening disease, not only in India, it throughout the globe, and at least 5 different groups of plant viruses that is infecting citrus. This is called as the citrus virology. Now the question is why this subject is so important. That is the first question. Because all these pathogens, these are transmitted by vegetative propagation. Vegetative means if the mother plant is infected, automatically progeny planting matter will be infected. That is first. Second point, most of these pathogens are efficiently transmitted in the field condition by different insect vectors. Third is, against this pathogen, virtually there is no chemical control measure is available, unlike fungal or bacterial diseases or nematode diseases, where some chemical measures to control these groups of pathogen like fungus, bacteria, nematodes is available. But this is not true in case of this group of pathogen like citrus virus and virus-like pathogen, against which there is no control measures are available. And most importantly, these pathogens, group of pathogens, few of the pathogens have very, very serious damaging potential. In any plant, in infected plant in any region or in a country, it may cause any of these pathogens, it may cause havoc to any part of the country in the citrus industry. As far as India is concerned, around 16 such pathogens have been reported to infect the citrus crop, among which citrus T-Stigma virus, citrus greening bacterium, citrus yellow mosaic virus, citrus exocotis viroid, and Indian citrus ring spot virus. These are the 5 target pathogens under this group, that is graft transmissible pathogens, virus and virus-like pathogens. These are the 5 target pathogens as far as the citrus industry of our country is concerned. I will briefly explain about what in our lab in Nagpur, that is Central Citrus Research Institute lab, what we are working as far as developing sensitive molecular diagnostics against this target pathogen is concerned, its characterization of this pathogen, and finally application of these diagnostic techniques in implementing citrus birdwood certification program. These are the areas we will be speaking today. Now this is quickly I want to tell you how these pathogens are being multiplied, being spread in the field condition. Citrus, vegetatively propagated crop, if the mother plant from which the cyan material is being taken, if that particular plant is infected by one or the other pathogen, automatically progeny planting material will be infected. Farmers purchase these plants from the nursery, whether it is a private nursery or government nurseries, without knowing the pathogen status of this planting material. They take them to the field, and once the plant is infected, within few years time, it is going to die, or it will give a poor yield. At the same time, this infected plant will act as a source of infection, source of inoculum for spread to the nearby healthy plant in the existing orchard. And this pathogen will spread from infected plant to healthy plant either by insect vectors or by mechanical means or by human being alone. This becomes the problem more severe, more complex. And you can imagine a country like ours where there is a need of about 150 lakhs planting material every year for new plantation as well as replanting within the existing orchard in the country level. Until and unless we implement citrus biodiversity certification program, the quality of this planting material, there is always remains a doubt about the quality, and that's what it is happening. There is a need for developing diagnostics. Now the question is, why? What is the need for developing diagnostics? Recent COVID, because of the COVID infection in the global level, even a common man, they are familiar about the detection of the COVID pathogen, real-time PCR, RT-PCR, these are the things. That has helped we people to understand that what is the need for developing diagnostics against the group of pathogens which is infecting citrus crop. This ensures crop biosecurity from invasive pathogens. Also helps to understand the etiology or the type of virus that is involved to cause an infection. Then also, once the diagnostic technique is developed, that helps in production of disease planting material, in export of our plant or plant parts, to understand the evolutionary background of the pathogen, and to understand the disease development process in the infected plant. There are many techniques that is being used by the plant pathologists, plant virologists throughout the globe as far as the detection of the pathogen is concerned. Few of the conventional diagnostic tools includes the field diagnosis based on the experience and the symptoms available or showing in the infected plant. We can guess that is the first level of diagnosis, that is field diagnosis. Where it is not possible, then we need to take the samples, coming back to the lab condition. For different conventional techniques includes is biological indexing. Then where the indicator plants, the set of indicator plants are being used to detect a pathogen. Then understanding the changes in the plant chemistry that is being caused by the causal pathogen, culturing of the pathogen, then virus induced structures that can be seen by right microscope in the infected plant, or directly seeing the virus particle under electron microscope. These are the different conventional techniques used by the plant virologists throughout the globe. But it has certain limitations. These conventional techniques, there is variability of symptoms, asymptomatic infection, low virus to the infected plant, but many times there is an interference by inhibitory substances or other pathogen that hampers the proper diagnosis of the associated pathogen. There are different molecular biological tools or techniques that is being used mostly by different labs in many countries, including ours. There are certain techniques like protein-based techniques. You are familiar with that because these are the things that has been taught in your class by your teachers. Protein-based techniques, whether it is ELISA or dot immunobinding assay or Western blot, these are the techniques that is based on the protein properties, whether it is spore protein or any other protein of the pathogen. Similarly, nucleic acid based different techniques, whether it is PCR or real-time PCR or LAMP, or recent last couple of years time, this is a LAMP, RPA and LFA. These are the techniques that are now these techniques are mostly used by different labs, including real-time PCR techniques. So these are the nucleic acid based techniques, and combining protein and nucleic acid, that is immunocapture PCR technique, which combines both protein as well as nucleic acid in the detection process. So these are the molecular diagnostic tools being used to detect a pathogen that infects the plant, not only citrus, any other plant or any other combination. So these are the techniques. But all these techniques has its own advantages and disadvantages or limitations. For example, biological indexing, biodiagnosis. Here, graft inoculation on indicator citrus plant, that has to be done. And just like lock and key plant pathogen or virus which is infecting, for example, citrus disticha virus. If a plant is infected or suspected to be infected by CTB, the respective indicator plant is acylene, that is Citrus olentifolia. We need to raise the indicator plant, then take the suspected field samples, graft inoculated in the screen house with the seedlings of the indicator plant, and then we have to wait for a few months' time. It may be 2 months to 3 months. In some combinations, the waiting period may be even more than a year. Again, that depends on the pathogen and the indicator plant. That combination is dependent, that is by indication. And the specificity is low to moderate, but this is very, very important. Then sap inoculation on herbaceous, again, is another biological assay where different herbaceous plants based on the pathogen and the combination. It depends on different that also. It takes relatively less time once we are using herbaceous plant, but here also the specificity is low. Light and electron microscope. There it is a moderate specificity, it takes around 60 minutes time. Serological techniques, protein-based techniques, within 24 hours we can get that, it's high. And nucleic acid based technique, it is again very, very sensitive technique, and its sensitivity is very high. These are the different diagnostic techniques being used by plant pathologists in many labs to detect the pathogen, the viruses, or any pathogen, virus-like pathogen, not only in citrus but also in other crops. As far as citrus is concerned, based on our survey, sample collections, and analysis of the samples after bringing them in our lab, we have come to the conclusion that so these are the areas. This is the northwest region of our country, where Quinoa mandarin is the predominant citrus cultivar in that region, where ring spot is the most common pathogen, followed by exocotis, cystic, and greening. Similarly, in the central part of India, these are the pathogen which is of serious nature. Similarly, southern part, citrus yellow mosaic, CTP, exocotis, greening, these are the pathogens which is of a concern in that region, because they are in the different regions, crop is different. Similarly, environment is different, and pathogen occurrence is also different. For example, in this northeast, ring spot, Indian citrus ring spot is the most predominant pathogen virus in that position. But this is not present mostly or very limited occurrence in other growing parts. Similarly, in southern part of our country, citrus yellow mosaic is a major viruses, mostly confined in that region, but in other places it is very less or negligible. As far as the diagnostics is concerned, symptom notice is very, very important in the field condition. Once you are working a particular crop for quite a reasonably good amount of time, you'll be familiar with different types of symptoms that is being developed by the pathogen. Here there are three different things. One is the which crop we are dealing. A particular pathogen will cause or causes different types of symptoms in whether it is a mandarin or it is a sweet orange any group or in acylene. Pathogen remains the same, but depending on the crop, the symptom expression varies. Similarly, different types of pathogen produces different types of symptoms. Third and most important is the symptom expression varies on temperature. Certain pathogen, they love low temperature. They are low temperature loving pathogens. For example, disticha, ring spot, and greening. They produce characteristic excellent quality of symptoms when the temperature is low. But for example, exocotis or phytoplasma, they are warm temperature loving pathogen. So their symptom expression will be much more prominent when the temperature is relatively high. So this is certainly, these are the distinct symptoms. Many a times you may not find classical symptoms in the field condition, there may be some confusion, there may be the infection of other pathogen in the field condition, but in those cases there is a need to take the samples in the laboratory condition and go for testing. These are the yellow background. These are the pathogens present in our country, and it has an efficient vectors being spread in the field condition. In the red background, just to give you an idea, that red background, these are the diseases which is very, very serious graft transmissible pathogens. Fortunately, these diseases are not present in our country at the moment, but these are present in other countries, and it is again being transmitted very efficiently by different insect vectors. Why I am showing this pathogen? Because many times we find that in last 5 to 10 years there is an effort by different state governments or state agriculture departments to import different citrus cultivars, exotic citrus cultivars for foreign countries, so that the basket of the different citrus varieties, fruits available to the consumers will be much more, and it can be fresh fruits can be available in the different years, different months of the country. That's of a year. That is fine, but we must keep it in mind while introducing new citrus cultivars, new citrus germplasms in the country. We must see that we are not introducing any new pathogen along with the introduced citrus germplasms. So this is biological indexing. You see, as I told you, indicator plants, and there is a say for indicator pathogen and indicator plants, and in that case we have to take samples, and then we have to go for the take the samples from the field condition, go for sample inoculation by different means, and only we have to wait for the symptom expression. This is biological indexing. Another one is that electron microscopy, directly seeing the virus particle under electron microscope. You can see these are the different types of particles. This is CTB rings, but these are the particles. Directly seeing the, but this is not visible in each and every lab. Only bigger national institutes like IRI, IHI, or even in your university also, this electron microscopy is available. But many times we need to take the help from other institutes to do the things, to see the virus particle under the electron microscope. So PCR, again, PCR based diagnostics, whether it is simplex PCR, this is a very, very common technique. Simplex PCR, we need to design the primer, target the pathogen site which is targeted, and then it has to be amplified by PCR technique, and we can see the virus specific band. Duplex PCR is again CTB, where this is an example where citrus tisticha virus is an RNA virus, citrus is a DNA virus. Both the viruses can be detected by duplex PCR technique. This technique we have standardized and published it. Purification of the virus and production of polyclonal antibodies by immunizing rabbit and develop the antibodies. That is again very, very important aspect. But many times what happens, especially for plexus viruses like CTB, purification is very challenging, and many times it also contains the host proteins. So that hampers the production of quality antibodies. So here what we have done, that PCR amplified product of the particular code protein, that particular amplified portion can be used for cloning and expression, and then the expressed code protein can be used as antigen to immunize rabbit and to produce recombinant polyclonal antibodies that we have produced, and that can be used successfully for detection of the virus particle by different serological means. Quickly, I'll tell you about the different types of symptoms. Since CTB produces quick decline symptom, yellowing symptom is a general types of symptom. Not necessarily always it will be by CTB. There are many other factors that produces yellowing type symptom. In that case, we need to take the sample and go for the testing in the laboratory condition. In acid lime, vein corking and vein flaking, These are the two very classical characteristic symptoms produced by citrus disticha virus on acid line. Leaf cupping and stem pitting, again, is very, very classical symptom. That is, in the infected plant, if you remove the bark, then you can see the typical pitting symptom. Again, in mandarin, you may not find the characteristic classical symptom as you find in acid line, but this type of small upright bunches with small yellow leaves of Nagpur mandarin, many times we found this type of symptom because of CTB infection. And smalling of reduction is food size, quality, that is another symptoms that is being produced by citrus disticha virus. As I told you, PCR based diagnostic technique, real-time PCR based technique, these are the techniques being used. Apart from previously that is serological techniques, still serological ELISA based technique is also being used. But couple of recently we have developed a reverse transcription recombinase polymerase based isothermal amplification coupled with lateral flow immunochromatic assay. This technique, we have standardized this technique for sensitive detection of citrus disticha virus. This we have just last year we have published it in scientific report. This is another lamp-based diagnostic technique. It's again very, very popular, and in last 8 to 10 years time, this technique has been standardized for many pathogens. In our lab, we have developed this technique, standardized this technique for CTB, for citrus greening, and for Indian citrus ring spot virus. These are the lamp-based diagnostic techniques we have standardized to detect the pathogen, different pathogens, and that also is we have published this our work. Citrus yellow mosaic virus is a very, very important pathogen, especially in southern part of our country, in different crops. This pathogen causes different types of symptoms, and this is the virus particle under the electron microscope. Duplex PCR, as I told you earlier, CTB and citrus yellow mosaic, we have standardized this technique. And again, this is another technique where that NCM based diagnostic technique just 1 minute, I'll stop here. There is a call from ADG. I'll just take 1 minute. Then NCM based nucleic acid extraction, citrus yellow mosaic virus. So there is no need for liquid nitrogen. There is no need for your -20 or -80. This technique, just by using a pestle mortar, where we can just after grinding that extraction can be spotted on NCM membrane, and that you can keep it in room temperature for a few hours. And specifically in a remote place where there is not much facilities like PCR, real-time PCR, or gel electrophoresis, gel documentation system, in those remote places, this NCM membrane spotted with the suspected plant samples, that can be shipped to the place where there is the facilities of PCR machine. Next slide. Here we have the same using NCM based extraction process. Here we have developed duplex PCR technique where both mosaic and greening can be diagnosed together by using this NCM based extraction. Indian citrus ring spot virus is another important pathogen which is prevalent mostly in northwestern part, Punjab, Rajasthan, Haryana, in that region. This is Indo-Mandarin. This type of symptoms is the virus particle, and this pathogen can be widely being detected by RT-PCR technique. And you can see in the symbol, many times in the field condition, a particular plant can be infected by more than one viruses, whether it is T-Stiger, whether it is a ring spot, or it is a mosaic. So more than one pathogen is in nature, it is available. In those cases, the things becomes more complicated. For detection of the Indian citrus ring spot virus, we have standardized a sensitive reverse transcription LAMP-based diagnostic technique for detection of Indian citrus ring spot virus, and we have standardized, optimized the temperature, the time, and the specificity also. It is so specific that it detects only the Indian citrus ring spot virus. No other viruses can be detected in the standardized technique. And this information last year I published in the journal Plant Disease. One of my students, Amol Kokane, who is just finishing his PhD, Amol worked on this pathogen for his PhD program, and this we have published it in Plant Disease last year. Again, this is another work done by Amol Kokane, that we have developed this technique, that successful detection of Indian citrus ring spot virus by qPCR technique. This also this is a very, very efficient technique for the detection of the pathogen, because what I want to focus is here that Indian citrus virus ring spot virus is a low temperature loving pathogen. In the field condition, we have found that especially during January, February, these are the 2 months when the temperature is low in that region, there are distinct symptoms is visible on the infected plant. Once the temperature increases, even the same infected plant, they fail to show classical symptom. That is the time we really need sensitive, sophisticated diagnostic technique for detection of the pathogen. And this technique, this is RT-qPCR technique, that is very, very sensitive for detection of this pathogen. This citrus Exocotis virus is another important pathogen, moderately present throughout the country, and this is warm temperature loving pathogen. Once the rootstock is very, very susceptible, like Rangpur lime, you can see this type of classical symptom in Rangpur lime you can develop. Rough lemon, sometimes you may not see any visible symptom, but only mild bark spitting symptom will be there in rough lemon. But it's just in tripolate orange or in Rangpur lime, is a classical bark spitting symptom will be there only on the rootstocks, and cyan portion you may not see any visible this type of spitting symptom. The plant will show moderate decline symptom because of the infection of these rings Exocotis virus, and we have developed the PCR technique for detection of the pathogen. In these cases, that etroxytron is the indicator plant for detection of the Exocotis virus, and it produces severe leaf epinesty. Once this etroxytron plant is inoculated with the test sample, it will produce this type of severe leaf epinesty symptom. Sometimes, depending on the composition of the viroid, it may show mild leaf epinesty symptom in the indicator plant. Sometimes it is stem necrosis symptom, and many times there is a mixed infection of different viroids in the in situ infected plant. Citrus greening is another very, very important pathogen. Citrus greening, this is in our country, we are finding this pathogen for almost last 70 years. But this pathogen has caused very, very severe damage to the world citrus industry, especially in case of US, in Brazil. This type of symptom we can find in throughout the world. Different types of symptoms in the infected tree, infected branches, infected leaf, infected fruits, and this is a classical leaf mottling symptom you can see in mandarin or sweeter. This type of mottling symptom in Nagpur mandarin. Many times the symptom is confused with zinc deficiency symptoms, mottling symptoms, zinc deficiency. In that case, we have to use the developed diagnostic techniques for proper diagnosis of the pathogen involved. In our lab, we have developed again recombinant polymerase based isothermal amplification combined with lateral flow assay. We have developed this technique for detection of this candidatus Dibirobacter asiaticus that causes citrus greening disease, and this information we have published recently in the journal PLOS 1. We have also developed standardized TACMAN real-time PCR detection technique. This is a very, very sensitive technique through which we can detect the pathogen candidatus Dibirobacter asiaticus not only in the infected plants but also in the citrus psyllid vector that transmits the pathogen from disease plant to the infected plant. So this technique is very, very sensitive, and we are using for different our other experiments. Similarly, lamp-based diagnostic technique also we have standardized. Lamp-based diagnostic technique for detection of greening pathogen, Candidiasis Liberibacter asiaticus, both in citrus and as well as citrus psyllid vector, diaphragm acid, we have developed this technique. And all these techniques, they don't need any sophisticated equipment. Of course, it has certain limitations because this technique is prone to contamination, but it does not need any sophisticated equipment like PCR, real-time PCR, gel documentation system. It does not, or gel electrophoresis, with more expertise and practice. These are the techniques can be used even in a very resource-less areas, but with technique. Now I'll talk. This is all about our developing diagnostic techniques. Now I'll speak a little bit about the characterization of pathogen, of different pathogens. It is not possible to speak all our work which we have done as far as the characterization of all pathogens infecting citrus is concerned. I am not doing that, but just to give you a few examples of the characterization of at least one pathogen, so that you can see that similar work also we are doing in other pathogens. For example, you can see citrus greening depending on the host, depending on the isolate, different types of symptoms. The citrus greening causing bacterium Candidiasis Liberibacter asiaticus, that it produces this type of symptoms. But initially, we are targeting only the 16S as well as 16S 23S, that proportion, to characterize the virus. And based on our observation that these two regions, 16S as well as 16S 23S ribosomal RNA, these are the two we can detect up to species level, Candidiasis Liberibacter asiaticus. But if you want to characterize the pathogen beyond species level, these two regions is not enough to characterize the pathogen. This information we have published in 2013 in the journal Phytoparasitica, that is being published from Israel. After all, we found that what next within the different species, what is the other, how to go for the further characterization of the pathogen. Then we targeted that other region, that is Klebsiella region. That region, that is Klebsiella, that is a number of tandem repeats, that is these three nucleotides present in a particular region of the citrus greening bacterium genome. Based on that, we have classified, we have isolated around 81 greening isolates collected from different parts of the country, infecting 8 different citrus cultivars. And based on the number of tandem repeats available in that region, hypervariable region, that we have classified in class 1, class 2, class 3, and class 4. Class 4 means in that particular region, which is around 650 amplified product, that is TRN number is less than 5. Class 2 means 5 to 10, that number. And class 3 means 10 to 15, and class 4 is more than 15. So based on that, we have classified that Indian isolates of Candidiasis Liberibacter asiaticus, and in 2015, this information we have published in the journal Phytopathology, being published by American Phytopathological Society. In fact, this is our first paper in the journal, prestigious journal, that is Phytopathology, being published from and in the global level, this is the second article. One first paper was published by a Chinese worker, and this is in the global level. This is our second part of publication in the world level on this aspect. This is the detailed publications which I have just spoken, the tandem repeat variability in genome locus, in a genome locus. Recently, last year, similarly, in this case, we have collected different isolates from Bhutan. That detection and molecular characterization of Candidiasis Liberibacter asiaticus, also in citrus disticha virus, which is associated with the citrus decline in Bhutan. And based on that, we have characterized both CTB as well as citrus greening bacteria. We have characterized that in case of greening, 4 different 16S RNA, Rufus gene, 15S ribosomal, these are the gene, and tandem repeats in cleavage 0 or 6. These are the 4 different regions of the genome of the pathogen we studied, and based on that, we have analyzed that this Bhutan isolates, this is classified into class 2 and class 3 based on this cleavage here, this region, and analogous to Indian isolates, and term A based on this region, 2 different regions based on that detailed molecular characterization, both for CTB as well as citrus greening bacterium present in Bhutan we have done, and this again we have published in 2021 in the journal Phytopathology, being published from America. Citrus witches bloom is another important pathogen caused by citrus phytoplasma. For this, we have standardized nested PCR technique through which we can detect the pathogen very quickly, very sensitive way, reliable diagnostic technique we have standardized. These are the pathogens which is not present, I told you in previous slide, the exotic pathogen present in other countries. Fortunately, at the moment, this pathogens is not present in our country. There is a need, because since our country, we are importing many citrus varieties from other countries, there is a need for developing international collaboration with the person, scientists working in other countries where this pathogen is present, so that in our country, we should be equipped with the developed diagnostic tools, diagnostic kit to detect this pathogen for accidental introduction along with the imported planting material. There is a need for collaboration. As far as the detection management is concerned, as I told you, that this group of pathogen is really very difficult to control, because in many cases there is no resistant commercial cultivar is available. In most of the cases, there is no chemical control is available. Any chemical which targets the pathogen that has effect on the plant as well. So there is virtually no chemical control is available for this group of pathogens. And most importantly, many of this group of pathogens, I am giving an example, of course, greening as a citrus greening as a model, but in many crops like Tistija or greening, it is severe, is very highly efficiently transmitted by different insect vectors in the field condition. So this is the challenge as far as the management is concerned. But we do have the composition strategies available. For example, production of disease-free planting material, about which I will speak in our subsequent slides. Then vector control, because in the field condition, different insect vectors, whether it is aphid for CTB or citrusilla for citrus greening, these are the vectors are being effectively spreading the disease. So we need to protect the crop from the infestation of different insect pests by going for chemical spray, especially when there is a young plants, or as well as different biopesticides or biological control of insect pests. These are the conventional strategies that is being used throughout the world. As far as the biotechnological approaches is concerned, these are of course mostly in a research stage, not in a very commercial stage at the moment, but these are the 3 areas: antimicrobial proteins and peptides, in silico inhibitor drug resistance designing, and genetically modified crops. In fact, these are the 3 areas our lab we are working, and we are working in collaboration with in different projects funded by DBT and DST Government of India, with our collaborators like Indian Institute of Technology, Roorkee. We have collaboration with IIT Roorkee. We have collaboration with University of Central Florida, and these are the three areas we are working at the moment, and we have got a lot of information. RNA is another biotechnological approach being used or being attempted by different labs in foreign countries, but in our country we are not working in that. But these are the areas. I will give couple of small examples afterwards. These are the areas we are working. Integrated management for winning the battle, whether it is a quarantine, it is a regulation, biodiversity certification production, then different cultural practices. vector control, use of tolerance. These are the different approaches that should be used in combination in order to manage this group of pathogen infecting citrus, so that the damage caused by this pathogen is minimized, so that the farmers can get good harvest, at least reasonably high harvest, to sustain economically. So these are the techniques that has to be followed. Shoot tip grafting is another technique that is being used throughout the globe to clean the infected plant. Even if the horticulturally superior plant, it is infected by one or the other virus like pathogen. So there is a technique, that is shoot tip grafting technique, that is used by the institute, by our colleague, to clean the infected plant, so that by using this technique, even from the infected plant, virus-free planting material can be produced, and we are using this technique at the institute. As far as the citrus birdwood certification program, there are 3 different steps. First one is identification of candidate trees, which are horticulturally superior, means it has a good yield potential. In the field condition, we are identifying candidate trees, whether it is a mandarin or sadguri or sweet orange. So it has an horticulturally superior plants. It is giving regular yields, it is giving good quality fruits, but apparently it is free from any visible symptoms. We need to identify that plants in the field condition and take the samples to immediately test in our laboratory condition, where they have the expertise, where they have the technology available. So these mother plants we are identifying, it has to be identified, tested against the group of pathogen. For example, we have identified after a thorough survey, say 10,000 plants in the field condition. We have identified 100 plants in the field condition which are horticulturally superior, and by using our developed diagnostic tools, suppose you are getting say 30 plants which are really free from this target pathogen. So this 30 plants is our asset. We need to protect those plants in the field condition and take another set of the bud sticks in your station to develop protected field foundation block. And this protected foundation block should not be used for taking fruits, but this will be used only for taking virus-free cyan material. And then side by side raising the rootstocks, whether it is a rough level or rankuline, in sterile soil mixture condition, again in the insect-free condition, in the insect-free greenhouse condition, and then commercial. These are the 3 steps in this exercise. I have told you: identification of mother plant, testing, protecting the virus-free mother plants, and develop protection foundation block, develop the rootstock in a sterile condition, take the bud sticks from this protected mother block, go for commercial multiplication. And you'll appreciate this is the picture at our institute, and every year we are developing more than 3 lakhs planting material which is free from this pathogen every year. And you'll appreciate that in last more than 20 years, up to including this year, we have produced more than 45 lakhs planting material free from this pathogen. That has helped the institute to earn about 15 crore rupees resource to the institute. And there are a couple of things which we need to do, that cultural practices, cultural procedures we must to follow. Every after harvest, we recommend that the plant has to be dead weed has to be removed. And you know that only after the harvest pruning, that pruning tools has to be disinfected by sodium hypochlorite solution, so that there is the spread of the disease from disease plant to infected healthy plant, it should be stopped. So there is a need for disinfection of pruning tools by using sodium hypochlorite solution. In the nursery condition, couple of more things we need to understand, that while we are transferring the seedlings, rootstock seedlings, from primary nursery to secondary nursery, we need to discard this type of wrong rootstock system. We need to take straight rootstocks while transferring from primary nursery to secondary. Another wrong method is, while we are shifting these proper rootstocks plants in the poly bag, having the sterilized rootstock soil mixture, having sand, soil, and FYM, we should not use finger, because this makes only a few inches, but the size is almost 8 to 10 inches. So we need to use the wooden stick to make a thorough hole from this to this, and then only we should use it this directly. And while shifting from primary nursery to secondary nursery in polywing bag, these rootstocks also need to be treated with fungicides. That is very, very important. Another thing, that while if we do not discard this plant, this type of bent root system in the nursery, in the long run, this will create problem like this, bent root system. Even if the root scion material is free from the viruses, there is no virus in the scion. But if you are using this bent root system in the rootstock, even in the field plant, even after 4 years, 5 years, there will not be proper growth. Plant will remain stunted, yellowing symptom. So it will be confusing with virus symptom or any other. But this is because of the wrong root system. So this has to be avoided in the beginning. Another thing, in most of the cases, what we do conventionally, as in the grafting, rootstock is Raplevon or Rangpur. We have taken the scion material and we have grafted it here. And immediately after the successful graft germination, we immediately cut this plant and we allow the scion to go. What I suggest that let us not cut it in this stage. Why I'm telling you, I have seen this in Florida, what they are doing. You see, this is the rootstock is typhoid orange or cariopsis range. And immediately after grafting, they are not removing the rootstock, top portion of the rootstock. They are bending it, the rootstock top portion, and so that there is more photosynthetic activity, more food production in the plant, and this by bending, this is allowing the scion to go straight. And just 2 weeks or 3 weeks before the final delivery, they are removing this rootstock portion. So I urge the people, if any nursery wants to convey this message, that immediately after successful, don't remove it, allow the rootstock like bend like this, and only 2 weeks before the plant, if you remove this, and afterwards, then you just 2 weeks before the plant. This is the picture we have seen in Florida nursery, an excellent growth condition of the disease-free planting material. And this I have already told that we have already distributed the planting material, and there is a need to replicate what we are doing at the Institute Nagpur. I know in some institutes this is being done, and there is a need to replicate the same birdwood certification program in all the citrus growing belts of the country, so that the citrus growers of different citrus growing states and regions, they can have the access to quality planting material. I'll be going a little bit, few more slides are there. This is another new area. We are lab in collaboration with IIT Roorkee as well as University of Central Florida we have done. This is an example where we have done the 2S albumin protein obtained from pumpkin seeds, around 12.5 kilodalton protein obtained from pumpkin seeds. We have isolated, purified this in combination of nano zinc oxide. This combination, we have found that this combination, it has an excellent effect on reduction on the inoculum load in the infected plant. And this information in the art we have demonstrated up to 4 months in the screen house. Right now we are doing the same experiment in the field condition, and this information we have published few years back in the world famous journal, that is PLOS ONE. And at the same time, this technology we institute in collaboration with IIT Roorkee and University of Florida, we have obtained the US patent. Now what next? This is a future line of work that I suggest and always propose, that there is a need to refinement of, still there is a need for further diagnostic techniques, there is a need for refinement, so that in right now we are detecting 2 pathogens in a single sort. Can we think of detecting or standardizing the technique where by single sort 5 major pathogens of citrus infecting in our country that can be detected? Using bioinformatic tools, we need to go for further molecular characterization of more number of isolates of our target pathogens from our country. As I told you, the insect vectors, what are the insect vectors or biotypes Is there any new insect vectors? Is there any new biotypes which we do not know at present? So that is responsible for spreading of the pathogen in the field condition. And again, there is a need to implement BADU certification program in the entire country in a Mason mode approach. Exotic pathogen approach, there is a need for international collaboration so that we do not allow the entrance of new pathogen along with the imported planting material. Identification of miles import cost protection, and then again there is a beneficial effect of a viroid for high anticipation as well as for solving the flowering problem. These are the, and then again finally the virus resistant transgenic plant. In this case, I want to share one more thing, that as I told you recently in the previous slide, the 2S albumin protein that is obtained from pumpkin seeds, that is successful. It has an inhibitory effect on citrus greening disease in infected plant. Now our next target is whether this plant origin gene, 2S albumin protein gene, whether can you use it to develop transgenic citrus plant that express 2S albumin protein gene. That is another area of work, and we are already working on that. This is my last slide. This is greening as a model, and this is true for any other pathogen, for any in citrus or any other crop. There is a host, susceptible host, efficient insect vectors, and the pathogen. In this combination, citrus, any crop varieties of citrus that is susceptible to this pathogen, efficiently transmitted by Citrusilla insect vectors, and the pathogen is Candidatus bacteria. These are the different. Now the target is to any of these by intervention, by scientific intervention and research, by enhancing the host resistance, so that either the vector or the pathogen, they cannot come here. Pathogen directly it will not come, or the insect vectors, that is also. So we need to enhance the immunity of the host by using different biotechnological approaches, so that the host is resistant against the pathogen or resistant against the vector. Similarly, there is an efficient vector. Can we think of controlling this by using pheromone trap, by biological control, insect sterility technology, or RNAi technology? These are the different areas of research through which we can manage the vector. If we manage the vector, if we can make the vector inefficient, then the spread of the pathogen from disease plant to healthy plant will be minimized. And lastly, targeting directly by pathogen. This is an area we are working. As I told you that we are working in this aspect where we are directly targeting the pathogen, because this pathogen is highly localized in phloem of the infected plant. Also, this pathogen multiplies in the insect saliva. So these are the area we are working in collaboration with our colleagues. So in the long run, probably down the line, maybe after 5 or 10 years, this is a model we visualize that in case of Citrusilla, this Muria plant, this is highly attractive or for Citrusilla. So Citrusilla infects or infests the Muria plant. Can we think of developing an RNA using the RNA tank, this Muria plant with RNA plus? So once the Citrusilla comes in as a border plant, automatically this Citrusilla will be malformed, defunct. It cannot move back, because this plant will remain as a poison to this insectilla. Because this Muria plant, it is very, very attractive for this Silla as a border few plants. Similarly, inside the citrus plants will be there. This can be a natural plant, normal without transgenic plant, or few plants. It can be transferred with the 2S albumin protein gene or the guava plant, which emits this beta caryophyllene. Guava plants as an intercrop is very successful in countries like Vietnam, where the guava plant as an intercrop, that volatile sulfur compound, that is beta caryophyllene, that works as an repellent against this Citrusilla. So can we think of developing transgenic plant that emits this volatile plant, so that the insects will not come? And thirdly, intercrop like soybean, gourd leaf, or Silla, that the plant itself releases some volatile substances that stops the visit of that discourages the visit of the insects. So this is a hypothetical question. I'm very sure the world, throughout the world, the research workers working on that, and down the line, will certainly, the scientific community will certainly come up with solutions to solve the problems of different challenges being caused by different citrus pathogens. In our lab, along with our own work, we also organize different types of training program for the participants, international clients. Meanwhile, we have organized, as Dr. Ramanna told in the very beginning, we have organized international training program for the participants from Bhutan, from Nepal, from Oman, and all this training program, this is our lab. Okay, all this training program are funded by respective government. Similarly, we have also organized training program for our different clients from our country, whether it is a state agriculture universities or KVKs, or 7 days or 15 days training program. We also organize in our lab in Nagpur, in a plant biology lab in Nagpur. And these are the people. I appreciate, I value the contributions made by all my team. These are the people starting from Balaji. He did his PhD working in our lab, right, last 12 years. He's working in US in a very big position. Dr. Balaji Aglebe, he worked in our lab on CTB, and last 12 years he is in US in a very good position. Then Sumit, he also did his PhD working with us. Dr. Sumit Bose, last 5 years he's working in Bangalore in a very good company. He's working and doing excellent. Similarly, Ashish, Manali, they have also finished her PhD. Ashish, after finishing her PhD, Manali was for 1 year in US to do her postdoc. Last year he has returned back, and she is also working in a very good private company in Bangalore. Ashish, after finishing his PhD from our lab, is working as an associate professor in Madhya Pradesh. And these are the people. Sunil and Amol, they will be submitting their PhD thesis in just few weeks' time. And I'm pretty sure both Sunil and Amol will go to US for their PhD, for their postdoc. And presently Mugendo and Datta and one more person has joined in last week. Presently, these 5 people are working in our lab. And also I recognize the valuable contributions and support I received from my collaborators, from IIT Roorkee Professor A.K. Sharma. He is the head of the biotechnology division in IIT Roorkee. And last 12 years, I'm working with Dr. Sharma. A lot of funds we got from DBT, DST, and again, Professor Swadesh Mukul Santa. He is the expert, he is world authority as far as nanoscience is concerned. This is a very clear message to all my colleagues, especially those who are new in the system. There is a need to have a team. Alone, not much can be achieved in research. There is always a need to have collaboration, building a good team with good understanding. If you progress, you can really contribute a lot in science. With this, I am through. Thank you very much. Thank you very much. Dr. Ramanna Sahib, I have finished my presentation. Dr. Ramanna, it is not coming. You have to unmute it.
Yeah. Madam, please take the program forward.
Thank you, sir. Thank you, sir. This is Dr. Swarajya Lakshmi, Professor Horticulture from College of Horticulture, Anantarajpeta. It's a very elaborative, informative lecture. We are very fortunate to have Dr. Dilip.K.Gosh sir on this platform for giving elaborate information on different bioagents that are causing diseases in citrus, and what is the importance of virus and how to detect virus by different means, field diagnostic methods and laboratory techniques, and latest advanced techniques in diagnosis of virus diseases, and how these can be multiplied and symptoms that are expressed in different plant species in different ways. All this information that is enriched the knowledge of our student participants in this lecture, sir. Now I request students or faculty, if any or having any interaction, if they want to interact with sir, it's a very good opportunity for all of us, sir. We are very, very thankful to you, sir, for sparing your valuable time and providing us with this excellent lecture, sir.
So it's my pleasure, madam. Thank you very much.
Advanced techniques of very, very nice, sir. Thank you, sir. These are important, sir. Rapid diagnostic method.
Thank you very much, sir, for an excellent. The lecture is a very highly informative lecture, sir. Not only for the students, but also for the other participants of today's program. It was really very impressive lecture, sir. I think this is going to be the best lecture in this 30 series of lecture program. I once again thank you, sir, on behalf of Honorable Vice Chancellor Dr. YSRHU, all the university officers, faculty and students, those who have participated in this program. Are there any, if there are any questions, sir, I think quickly, any queries from the students and staff?
Good evening, sir. This is Ruth at COH Anantrajpet, sir, in the Department of Plant Pathology. Citrus greening is a major disease in Andhra Pradesh, sir. Is there any this advanced microbial or advanced nutritional solutions against citrus greening disease for controlling this disease, sir? And also, is there any bioagents used for controlling of citrus greening, sir? You already mentioned that nanotechnology for management of citrus greening disease, sir. Is there any information for control of this greening disease in Andhra Pradesh, sir?
So thank you, madam. First thing is that greening is a problem not only in India, it is a global problem. If you see the literature, that 2005, the disease first came to Brazil, US and Brazil. Uske baad na, after that, it made a huge loss to both the industry of US as well as Brazil. So the whole world is facing this problem and working on controlling this. But whatever available solutions are there, first one is that production of disease-filled pantry material, insect vectors. And your first question was nutrient management. It's a very, very important point. Because once even the plant is infected, so there is no direct chemical control. Antibiotic cannot be used. There is no chemical control. One of the best way to enhance the longevity of the orchard is that to give proper nutrition, so that at least the plant, which otherwise would be died in 5 years' time, by proper nutrition, micronutrient, then at least it can, the longevity of the orchard can be enhanced. Nutrition management is very, very important to enhance the longevity of the orchard. Second, second your question as the other I have already spoken, other methods that control of insect vectors, then there is no direct chemical control. Other aspects मैंने जो discuss किया, the most of the things are in a very researchable stage, but at the same time you can think of using that guava plants intercrop. There is no direct control, but once you are planting guava, this is demonstrated in other countries. It releases volatile sulfur compound that stops the visit of the psyllid. Means visit psylla नहीं आएंगे तो आपको inspection come. That is indirect way of control. If you go for other crops like soybean or in that is also some releases some volatile compound, but these are the indirect method of controlling that. Proper nutrition, disease-filled pantry material, insect control, And even if plant is seriously infected, then you need to remove it so that inoculum load is reduced. These are the areas you have to follow.
Thank you, sir. This is very useful for our students in the detection of plant viruses in citrus crops, sir. Thank you, sir.
So thank you very much, Dr. Ramannaji and Dr. Srinivasaluji and Madam. And thank you very much for giving me this opportunity. I have, as you told, I've tried my best to share what I have learned with you. And on my behalf, I once again invite all of you, whenever the situation improves, you feel free to visit our institute. Thank you very much.
Thank you, sir. Thank you, sir. Very
Thank you,
nice
sir. Thank you.
lecture, sir.
Thank you, sir. Now I take it as a privilege to say formal vote of thanks on behalf of College of Agriculture, Anantarajpeta. I thank our Vice Chancellor, Sir, Dr. Janaki Ram, sir, for providing us the great opportunity to form this platform and meeting with eminent scientists from various central research stations, ICR organizations, for meeting and discussing about all the advances in citrus crop research activities going on in citrus crop. Now I extend my thanks to all university officers and all teaching faculty who are involved in organization of this program, this citrus graduation webinar sessions. And I extend my thanks to all student participants who are encouraging us by their active participation. And I extend my thanks to all co-conveners, and all from Citrus Research Station and HRS Anantarajpeta, Citrus Research Station Tirupati, who are encouraging us for conducting these events, these sessions regularly. Thank you, one and all. Thank you, sir. Ramana, sir. Thank you so much. Sir.
Madam, thank you, madam.
Thank you, Daksa. Bye bye. Take
Bye,
care.
bye, Doctor. Thank you.
Bye-bye.
Thank you, bye, sir.
Bye bye.
Bye.
Take care. Bye.
Bye, sir.